of Dr. Kanagesswari, over to you.
Thank you, Aisha, for the wonderful introduction. Good afternoon, everyone. First and foremost, thank you for attending today's session. So let me share my screen first. Can you see my slides?
Yes, can.
Okay, and so basically, as first and foremost, I would like to extend my gratitude to Institute for Medical Molecular Biology, Molecular Biotechnology, IMMB, UITM Sungai Buloh, for giving us this opportunity to present the talk today. And I thank all of the participants for attending today's session. So, as Aisha mentioned, today I'm going to give a talk titled Next Generation Sequencing, the Past, Present, and Future. So this overall talk is about just giving an idea about how the sequencing world has evolved over the past decades and what kind of application it has and beneficial to the medical research. So without any delay, let's get started. So for the presentation purpose, I will be turning off the video and I'll on it back for the Q&A session. So before we get into the talk for today, let me briefly introduce our company, TREECODE Sendirian Berhad. TREECODE was founded in 2012. We are 11 years old as of today. Our main business portfolio is to offer customized genomics solutions based on the advanced short and long read sequencing technologies. On some occasions, we also offer genome scaffolding technology. And since this year, we started offering metabolomic services to our clients. We believe that our accumulated range of knowledge and insights in the sequencing industry has helped us to help our client to bring their research to the next level. Over for the past 11 years, we have successfully consulted and delivered many cross-platform sequencing projects for both our local and international clients. On a side note, we also supply high-quality molecular biology reagents, kits, and laboratory equipment whenever such needs arises for our customers. This is an overview of today's presentation. First, I'll give an overview about how the sequencing world has evolved. In the second section, I'll be mainly talking about the applications of omics technology through various sequencing approach and some idea on how it can be beneficial to the medical research. And the third section would be just giving an overview about TREECODE's omics services. And last, the talk will end with the Q&A session. Now, let's talk a bit more about the sequencing worlds and its evolution throughout the several decades. Before we dive into further, let me just briefly talk about what is sequencing first of all. So sequencing is an approach involving using biological methods along with the advanced technology to obtain the nucleotide sequence, be it a DNA or RNA of an organism, where it allows us to explore and analyze the genetic information of a particular species in detail. So basically, we have the DNA and RNA in a cell, which comprises of different nucleotides. And we want to now take out this information out from the cell and put it into the computerized format where the codes, the 4-letter nucleotide codes are jumbled up, as you can see from here. So now from the cell, we took it out and made it as a data which is available in the computer. So now we have this data. What we gonna do? This is where bioinformatics comes in. So bioinformatics involves using computational tools to interpret this biological information of an organism based on the determined nucleotide sequence. So now, as I mentioned, the nucleotides have been taken out from a cell and they've been made as a puzzles, 4-letter codes. Now, bioinformatics allows us to put these puzzles into organized details, which will actually let us know about the biological information. And for this, we need to use different computational tools that are through scripts, or any statistical softwares available. Over the years, there has been remarkable progress in the omics technology spanning from as early as 1869 until today, where we have achieved to obtain the complete gapless sequence of the human genome. So in the next few slides, I'll be talking more about how the sequencing world has evolved. I just wanted to share this diagram just to give an idea about how much the sequencing world has evolved, and it will be continuing to evolve for many more years. This is a very short overview about how the sequencing world has evolved, and in my personal perspective, we can categorize the sequencing world evolution into 3 different generations, starting from first generation, which is more of performing short read and pioneer sequencing, followed by the second generation sequencing, where we also refer to them as the next generation sequencing. And last but not least, I would consider where we are right now, which is the third generation sequencing, where the sequencing world has evolved tremendously to bring the best benefits it can bring to any researchers who are focusing on the omics field. First generation sequencing is considered the breakthrough era. One of the biggest pioneer of sequencing in this era is the Sanger sequencing. And here, to make it very simplified about this sequencing itself, it's enable us to sequence a single sample, and from the single samples we are looking at a single sequence. The main characteristics of first generation sequencing, particularly Sanger sequencing, is that it is enabled to sequence out short reads, usually somewhere in the range between 1000 to 1500 base pairs, and the sequencing reads that obtained are of high nucleotide accuracy. Sanger sequencing mainly works on the technology of utilizing D-deoxynucleotide sequencing, and the nucleotide detections are done via capillary electrophoresis. This is the modernized version of Sanger sequencing. Used to be, this Sanger sequencing was done using gel electrophoresis method. One of the key characteristics about Sanger sequencing is that you would require unique primers to sequence out the single sequence from the single sample. Here I mention it as a limitation, but it's just a very important characteristic of Sanger sequencing, where it produces low throughput reads, which allows you to get the nucleotides off of a single sample, but still the sequencing accuracy is kept at a very highly accurate manner. One of the important achievements using Sanger sequencing is that we have managed to sequence out the first complete genome, FIX174 of bacteriophage, which is currently used as a DNA positive control in many of the second generation sequencing platforms, particularly in IMMB. The second generation sequencing, also known as the NGS, which became the revolutionized era, where short-read sequencing with high throughput data generation technology was introduced. Just to give an idea, this was somewhere in the early 90s, where one of the second generation sequencing platform was introduced, which using chiral sequencing, where fluorescent based nucleotides were utilized in the sequencing to detect and sequence out the single sample sequences. One of the key characteristics of this second generation sequencing is that we are allowed to sequence out multiple samples, and within the multiple samples, you can also sequence out multiple sequences. Another key characteristics of second generation sequencing or NGS is that you would rely on performing a lot of template amplifications to provide a better signal strength and high sequencing nucleotide accuracy of the samples. And these are some of the examples of the instruments available in the market. And each instrument is unique because they utilize their own platform-based sequencing technology and nucleotide detection approach. And also, just to give an idea about it, second generation sequencing is that the reads are also short read sequencing, but the read length is somewhere between 50 to 300 base pair, where the sequencing accuracy is relatively high and their attempt to be kept as comparable with Sanger sequencing. One of the main sequencing platforms widely used in the market is Illumina for short read sequencing technology for the NGS purpose. And this is also the platform we recommend to our clients for any short read sequencing service. Illumina utilizes sequencing by synthesis technology, and the read length are between 50 to 300 base pair. Illumina short read sequencing platforms allow for sequencing done as in a single end or pair end read modes. To keep it very layman term, single end reads or single end sequencing means that the sequencing is done from one direction, while pair end reads or pair end sequencing is done in both directions. Usually, we recommend Illumina's short read sequencing service for these particular services, such as when the clients are interested to perform whole genome sequencing, which is in through race sequencing mode, RNA sequencing, targeted sequencing, metagenomic sequencing, and epigenetic sequencing. The choice between single and pair end sequencing very much depends on your project needs, which we will usually advise our clients when we are having a discussion with them about their research projects. Now, the next evolution happened in sequencing world, where the third generation sequencing instruments were introduced into the market, where we consider as the unleashed power of NGS era. So what is the main differences between the second and third generation sequencing is that where instead of performing short read sequencing with the third generation sequencing platforms, you are enabled to perform long read sequencing, where the sequencing rates are ranging from as short as from 5 kb to 30 kb or even more. These are some of the platforms available in the market when it comes to third generation sequencing platform, such as the Pacific Biosciences PacBio, which is this instrument, and Oxford Nanopore Technology ONG, which are these instruments. Again, similarly to the second generation sequencing platforms, each of these sequencing instruments have their own unique platform-based sequencing and nucleotide detection approach. Again, similar to the second generation sequencing platforms, long read sequencing instruments are also allow us to sequence multiple samples in one go, and within the same sample, you can actually sequence out multiple sequences. One of the few characteristics about third generation sequencing is that you do not require to perform template amplifications, meaning that in third generation sequencing platforms or in these long read sequencing platforms, the sequences are done using single molecular sequencing and real-time sequencing, where researchers can obtain native information of our biological organisms by using these third generation sequencing platforms. So again, To our customer, most of the time we offer PacBio instrument as one of the long-read sequencing service approach. PacBio utilizes single molecular real-time SMRT sequencing technologies where it performs the SMRT sequencing in two modes. The first mode is known as continuous long-read sequencing mode, where the read length can go up to 175 kb, where any large repetitive region, transposable elements, and any large structural and indels can be easily identified. While the second mode is known as circular consensus sequencing mode, where the read length is still kept in a very long read length, such as around 20 kb. But what happens here is that each read is sequenced multiple times to improve the accuracy, where during this sequencing mode, PacBio can produce not just long reads, but reads with high sequencing accuracy up to 99.99%. We usually recommend PacBio services for researchers who are interested to perform de novo whole genome sequencing, where researchers are interested to assemble a native or a newly species genome. Researchers would like to perform RNA or isoform sequencing, again to obtain the transcriptome information of a particular organism, as there's no reference genome available for them. To perform epigenomic sequencing, I'll talk about this more in the later slides. To perform long-read amplicon sequencing, also to perform metagenomic sequencing. Again, depending on the discussions and what kind of objectives the researchers want to achieve at the end of the day, we will advise them accordingly whether to go with long-read sequencing or with short-read sequencing. Now, I have talked about the evolution of the sequencing world in the last few slides. And throughout the slides, I was keep talking about the read length itself, where the first and second generation sequencing are more focused on sequencing short reads, while the third generation sequencing is distinguished by performing long read sequencing. So this might raise the question, why in the first of all the sequencing world has evolved tremendously, and why one of the key factors they were focusing on the improving on the read length. So this is just to give an idea about why the evolution happened in a way where we are doing a direct comparison between short read sequencing against the long read sequencing. Imagine the biological organism's DNA or RNA as a puzzle. With short read sequencing, we are at the end of the day managed to have high sequencing accuracy, which allows us to obtain the biological information of an organism. But due to the limitation of the read length, most of the time, occasionally, researchers cannot fill in the gap between certain sequences such as the repetitive region, large indels or structural variants, as well as these transposable elements. So at the end of the day, you're still having a picture or full picture of the biological information of an organism, but there are gaps in between, showing that there are still information left behind. However, with long-read sequencing, because the read length is extremely long, it can span across the repetitive regions and such, allowing us to obtain more complete biological information of the species of interest. One of the disadvantages that was noticed earlier in the long-read sequencing is that its sequencing nucleotide accuracy was not at its best, but even that, thanks to the evolution and improvements made in the third generation sequencing platforms, nowadays, even with the long read sequencing approach, we are still getting long read length, but the sequencing nucleotide accuracy is kept as accurate as comparable with the short read sequencing platforms. So with the long read sequencing approach, you are having a more complete puzzles which allow us to perform subsequent research using this biological information, be it the DNA or RNA, at much more confidence. Now, this is an overall view how an NGS workflow looks like. So the first step is always extracting the nucleic acids, be it DNA or RNA. Once you obtain the DNA or RNA of an organism, you would undergo library preparation. So here, what happens is that unique adapters and index primers or such matters are added into the samples. This will allow the samples to be readily sequenced in any of the platform of choice. So once the library preparation is done, the samples are then sequenced in any sequencing platform of choice, be it Sanger, be it short-read sequencing platform, or be it long-read sequencing platform. Once the sequence is done, you obtain the sequencing data in computerized or digitized format. Then you can proceed with bioinformatic analysis. Now, when it comes to bioinformatic analysis itself, you have three different stages where we consider them as primary, secondary, tertiary analysis, where you first look into the quality of the data and filter them and edit them or adjust them to become high quality data. Then subsequently, you perform secondary analysis depending on what type of data you are working on and what is your research objective, and followed by again tertiary analysis, where again depending on your research objective and end goal, you will be performing specific analysis. And one of the key important analysis you'll be doing at the tertiary stage is actually performing a lot of statistical analysis. This is because whatever data you found which is biological significant, you need to ensure them they are also statistically significant for publication papers and for the reuse or re-experimental purpose. So when it comes to bioinformatic analysis itself, you can perform the analysis using command line tools where you have to, write several different scripts to run the analysis, or you can use web-based tools where these command line scripts are already made as options for you to just click with your mouse and perform the analysis at the end of the day. Again, whether you want to go using the bioinformatic analysis via the command line tools or web tools, it also depends on your research objective and how much you have the necessary training to perform the bioinformatic analysis. Before I move on to the next section, I just wanted to talk about one of the biggest achievements that has been achieved in the sequencing world as of today, where last year we have managed to obtain the first complete sequence of a human genome. So the Human Genome Project started around in 1990, and in 2003 the researchers have announced that they already have the human genome sequence completed, but the journey didn't stop there. Over the years, researchers were continuing trying to improve the human genome and trying to see, are we really have the best human genome or the complete genome in hand for the last several decades. So in the initial stage, we were roughly knew it was around 3 GB, the human genome. Thanks to the improvements and introduction of the long-read sequencing platform, both PacBio and IMMB, last year, we have managed to utilize these platforms to actually generate the complete genome sequence. With this genome, researchers have found that there are more unknown genetic and structural variations were present in the genome, which was previously unknown. Having this complete gap-free sequence of the human DNA become critical for researchers to understand population variations across the world, and it allows us to have better knowledge on how genetic variations in the human genome can contribute to different diseases in different populations. Let's move on to the second section of today, where I'll be giving an overview about applications of omics using these different sequencing approaches. Now, I was always talking about sequencing just now. Suddenly, I'm introducing this word, omics. So, what is omics? The dictionary term of omics is that anything, an overall comprehensive view of a particular component or molecules in a cell, depending on its category. So, in today's world, the omics world can be divided into four main categories, where genomics and epigenomics are focusing on the entire DNA content of an organism. Second category is transcriptomics, where the entire content of RNA in organism is explored. The third category is proteomics, where the protein content of a cell or organism is explored. Last but not least, metabolomics, where the metabolite content of an organism is explored. So at this moment, genomics, epigenomics, and transcriptomics, which is focusing on the DNA and RNA, are well studied and established omics, while proteomics and metabolomics is slowly gaining its momentum. For today's talk, I'll be mainly focusing on the genomics, epigenomics, and transcriptomics world. Now, let me give a bit of overview about how omics technologies, especially using different sequencing approach, plays a role in the genomics and epigenomics world. Whole genome sequencing approach allows researchers to determine the complete sequences of an organism genome, be it the chromosomal or nuclear DNA, mitochondrial DNA, chloroplast DNA, and any other organelle DNA. WGS is actually a very powerful tool for researchers who are interested to perform de novo whole genome sequencing, where the researchers just want to assemble a sequence of a genome or of an organism that has not been studied before, or to perform whole genome resequencing, where a reference genome is already available in the database for researchers to use that reference genome to perform sequencing of their particular sample and to look into genetic variations. So both de novo whole genome sequencing and resequencing allow us researchers to have a comprehensive understanding of different variants and the genetic information in a population. To add on, whole genome resequencing actually plays a crucial role in genome-wide assertion studies, where common genetic variations in different individuals in a population are characterized to determine their genetic association with specific phenotypes. So this is just an overall view how next generation sequencing playing in sequence in two different modes, be it race sequencing or de novo assembly, or known as de novo sequencing. So basically, during race sequencing purpose, most of the time the reads obtained are actually aligned against the reference genome, and we are mainly focusing on looking at different variants such as the copy number variants, structural variants, or variants such as the SNPs and indels. While in de novo sequencing, the researchers are using the sequenced reads to perform draft assembly. Basically, they want to obtain the genome of an organism, and if the situation permits and the research objective allows them, they can go up to chromosome level genome assembly, and having this complete genome, then they can move on to the next step where you are annotating the genome itself, where you want to obtain the information on the protein and non-protein coding region of a particular organism. This is whole genome sequencing itself is just looking into the entire genome of an organism. Now, in this category, whole exome sequencing, this approach is mainly allows researchers to focus on looking at the coding regions of a genome, which is usually comprises 1 to 2% of the genome in any organisms of interest. So earlier in WGS, we are focusing on the entire genome, but in WES, we are just focusing on the entire exome of an organism, which is only focusing on the coding region. WES is a very powerful tool when it comes to any medical research work because researchers found over the years a significant portion of the disease-causing mutations are estimated to be occurring around this exome region. So it is more beneficial for researchers to focus on looking at this region, and for that, WES plays a beneficial role. One of the key characteristics of WES is that the data required for performing this sequence is also 10 to 20 times less, but at the same time, significant SNPs and indels can be captured. So, like I mentioned just now, whole exome sequencing allows researchers to uncover important genetic variations that can influence heritable traits and phenotypes in conjunction with the organism's genotype. Particularly in this case, is very useful in clinical research. Now, the next approach is targeted sequencing. So as you can see from the first few slides, we are moving slowly into a very, very targeted purpose, starting from the genome, exome, and now we are looking at targeted sequencing. So targeted sequencing is a method where researchers can only focusing on certain parts or regions of interest in an organism without spending the money and effort to sequence out the entire genome of a sample. Again, this plays a very important role in a clinical research where the researchers already know what are they want to look at and they are just focusing on looking into that and performing further research. So to perform targeted sequencing, you can utilize two different approaches. The first one is amplicon enrichment-based targeted sequencing, where researchers are performing PCR amplifications of the regions of interest, sequence them, and perform data analysis. The second approach is hybridization capture-based targeted sequencing, where researchers are again focusing on the target region of their interest, but they are designing special probes to only capture those target regions and then perform sequencing and subsequently data analysis. So when it comes to clinical research or medical research, the targeted sequencing is very useful when focusing on very clinically important diseases such as cancer, reproductive diseases, metabolic and immune disorders, infectious and inherited diseases. So at this stage, most of the clinical researchers or diagnostic field tend to do more of targeted sequencing approach because they are very much focused on in the disease they are focusing, and they want to obtain the most specific information that will allow them to make important discoveries and subsequent research. Now, most of the approaches I was talking about earlier is actually focusing on a particular organism. Specifically, it can be an animal, it can be a human sample, or it can be plant, or even a particular microbe, bacteria, fungi, or virus. But there is another interesting approach that you can perform with sequencing, which is known as metagenomic sequencing. So metagenomic sequencing is a method that allows researchers to study the microbial community that obtained from a specific environment by performing functional gene screening or genomic sequencing analysis. So metagenomic sequencing is mainly performed using samples from examples such as the environment, such as the soil and water, or from clinical samples such as the fecal and gut. And it's heavily focused on exploring microbial diversity, community constitution, functional gene explorations, and also allow researchers to actually characterize the genetic and evolutionary relationships of these microbial communities, and last but not least, to look into the interaction and relationships of these microbial communities, be it with the environment or be it with the host samples. So when it comes to metagenomic sequencing, you can perform two different approach. The first one, what we call is amplicon metagenomics, where you are actually targeting a specific region such as the 16S rRNA gene to focus on a particular microbial community. So by performing amplicon metagenomics, either you are focusing on the bacterial microbial community or fungal or virus. So here the main purpose is you would love to have more understanding of the taxonomic compositions and the microbial community diversity in the particular sample you obtain from the environment. The second approach is shotgun metagenomics, where researchers are looking into the microbial community of environmental sample, but here they are more focusing on looking into the microbial community diversity using functional genes and pathways analysis. So here, instead of just focusing on the diversity itself, they are actually mainly focusing on the gene expression and functional potential of these microbial communities. Now, in the genomics and epigenomics world, as I mentioned earlier, so one of the approach you are taking is about performing epigenomic sequencing. What is epigenomics? Epigenomics is the study of looking into epigenetic changes such as chromatin structure, histone modification, and DNA protein interaction of an organism, where these modifications are affecting the gene expression of an organism, but without making any modifications in the DNA sequences. The two famous epigenetic processes that widely studied is the DNA methylation and histone modification. And thanks to the sequencing technologies, you can actually perform sequencing to obtain these two information out from a particular organism of interest. So when it comes to DNA methylation sequencing itself, you can actually perform whole genome bisulfite sequencing using the short-read sequencing platforms, where you're focusing on the methylated and unmethylated cytosines. While interestingly, in the long-read sequencing platforms, you can obtain the native methylation information of an organism during the whole genome sequencing itself, without performing any alterations to the samples of interest. The second step is performing chromatin inhibitor precipitation sequencing, also known as ChIP-seq. So you can perform ChIP-seq, which is usually done using short-read sequencing platforms, where you are more focusing on looking into the histone modifications of an organism in interest, where you're also mainly looking into the histone modifying enzymes of an organism, or looking into chromatin remodeling enzymes of an organism, where these two enzymes are playing crucial role when it comes to altering the chromatin structure of an organism, which subsequently going to affect the gene expression of the particular organism. Now, the second omics world I would like to talk about further in today's talk is transcriptomics, which is focusing on the RNA. Transcriptomic sequencing, or also widely known as RNA sequencing, allows researchers to perform high throughput sequencing to get comprehensive insights about the transcriptome of a cell. So compared to any of the previous research methods, the current NGS approach allows for customers or for researchers to have more enhanced coverage and resolutions of this transcriptome of a particular organism of interest, which allows them to have a better understanding of the gene expression of the particular organisms. So overall, RNA sequencing allows us, for one of the main characteristics or one of the few important benefits of performing transcriptome sequencings are to detect rare and novel transcripts. So these are regions that are not previously explored, to actually look into and quantifying the transcriptome present in an organism and look into differential gene expression. Interestingly, you can perform transcriptome sequencing in order to obtain information on the fusion genes and alternative splicing information. Similar to the first, these two approach, you also want to again obtain more information about the common transcript already known. And last but not least, you can also nowadays perform transcriptome sequencing to obtain information about the variants such as the SNPs and the indels. So transcriptome sequencing meaning is that you are looking into the entire transcriptome of cells, but under this transcriptome sequencing, you can actually perform very few different specific sequencing, which I'll be talking about now. So the first sequencing is known as mRNA sequencing. So in this approach, you are actually focusing on the coding RNA molecules known as the messenger RNA. So the main purpose of performing mRNA sequencing is actually you are focusing on the differential gene expression occurring in an organism when they are exposed to different treatments or in different conditions. And you want to understand how this gene expression is differentiated when they are, undergo different conditions throughout their developmental stage or such. So another one of the interesting approach you also can do is total RNA sequencing, or also known as whole transcriptome sequencing. So in this approach, your researchers are actually would like to know both information of the protein coding molecules as well as the non-coding RNA molecules. So during this approach, the customers are performing two different objectives. The first objective to understand the gene expressions of differentially expressed protein coding RNA molecules, but at the same time, they also, in the second objective, they also want to get more information about the gene regulation pathways of the particular organism. The third RNA sequencing that you can perform is also known as small RNA sequencing, where in this approaches, researchers are only focusing on small RNA molecules, which usually range between 18 to 30 nucleotides. These non-coding RNA molecules play a very important role when it comes to gene regulation pathways. So by performing only small RNA sequencing, the researcher is only focused to know what type of gene regulation pathway is this particular organisms that they are analyzing undergo. Now, let's talk more about meta-transcriptomic sequencing. So as I talked about all the previous slides, how metagenomic sequencing allows us to explore the microbial community of environmental or host sample, where we are focusing on the DNA. Meta-transcriptomic sequencing allows our researchers to look into the RNA sequences transcribed by the microbial community, be it in the environmental or the host samples. So this is a very interesting approach where you are trying to look into the functional gene pathways similar to the shotgun metagenomics, but you're actually more focused on the RNA. So one of the long-term application of metatranscriptomics is actually when you're working on host samples, you can actually have an idea about understanding the immune responses, be it in the microbial or in the host communities, to understand how the host is affected or how the microbial community is affected when they are in a host sample. This in the long term will allow us to make more specific decisions about discovering different drugs that can allow clinical researchers to target any of the harmful microbial community present in the host sample, at the same time to work on drug delivery system. This is just a short overview between metagenomics and metatranscriptomics. As I mentioned just now, metagenomics is mainly focusing on the DNA or genomics of a microbial community, while the metatranscriptomics is focusing on the transcriptomics information of a microbial DNA. This is very much focused on the DNA, while metatranscriptomics is focused on the transcribed DNA, which is mostly mRNA. Of course, the extraction procedure itself is specific whether you are extracting DNA or RNA, but ultimately, what is the purpose of performing the two different sequencing approach? With metagenomics, the researcher's main objective is to understand the microbial diversity of a microbial community, while in metatranscriptomics, the researcher is mainly aiming to understand the functional diversity of a microbial community. This entire slide is just to give an overview how these different omics world and the particular sequencing approach we can undertake will be useful in the medical research perspective. So performing various different sequencing approach using the various different sequencing technologies allows our researchers to have more understanding that will be useful in these different applications, such as human genomics, clinical diagnostics, pharmacogenomics, infectious disease genomics, cancer genomics, host microbiome interactions, and last but not least in epidemiology and population study. So overall, by performing all these different approaches, at the end of the day, the researchers can have powerful information or useful information about an organism, be it the host, which can be a clinical sample like human samples, or be it about the microbiome community itself, be it the bacteria, fungi, or virus. All of these informations are useful when it comes to clinical medical research setting, which will allow us to perform subsequent focused research. Before I end the session, let me just give a few examples of how these different sequencing approaches can be useful in the medical settings, where researchers in previous years have performed whole genome sequencing to characterize different bacterial pathogens from the host samples. The first one is to actually isolating and sequencing these Mycobacterium fortium samples to understand how they can become drug resistant, where the second paper is actually looking into Streptococcus pneumonia strain to understand how its characterization can affect its virulence and how much it can be harmful to the environment or to the clinical samples. Transcriptomic sequencing is also quite famous in the medical research setting, where researchers can perform either whole transcriptome sequencing or mRNA sequencing, where both approaches allows them to understand differential gene expression of a microbial species and look into how this differential expression of these microbial species can affect the host, or how are they affecting the microbial species itself. This section is just to give you an overview about TREECODE omics services. So in overall, TREECODE can provide any sequencing services beginning from whole genome sequencing up to metagenomics and metatranscriptome sequencing. We also offer customized sequencing solutions, where this is offered based on the discussion we have with our customers to understand their project needs and any subsequent requirement. So one of the ways sequencing world had evolved, and we are looking into new different approaches. So today I would like to talk a bit more about barcode tag sequencing, also known as BT-seq, which was introduced in the last 3 years, where we see this technology is very much useful and very innovative compared to all the different technologies I talked about, where it can be very much useful to the customers at the end of the day. We also provide bioinformatics services, and also for any researchers who like to set up their own bioinformatics lab, we also provide high performance computing services. Now, let me just briefly talk about barcode sequencing in further details. So, barcode tag sequencing, or BTSeq, is an NGS-based molecular barcoding technology where we believe it can be a suitable alternative to Sanger sequencing, but it also has more potential than that. BTSeq is very much useful to be used as a de novo or reference-based sequencing or resequencing, but the target samples are mainly of amplicon or plasmid samples. One of the key characteristics of BTSeq is that it allows researchers to sequence wide range of DNA sizes from 200 base pair to 20 kb. The sequencing accuracy is kept at a very high level where it is around 99.99%. One of the best part of BTSeq is that you can sequence unknown sequences without the need of any unique sequencing primers. And in the long term, it does become a cost-effective solution. So just to briefly give an overview about BTSeq is that once we have your PCR product and plasmids, during the library prep, Salamic's proprietary technology of enzymatic fragmentation is performed and unique molecular barcodes is added to the samples. Once the library preparation is complete, sequencing is performed using any of the short-read sequencing platforms, particularly using the Illumina platform. And during the bioinformatic analysis, the molecular barcodes attached during the initial library preparation is used as reference to stitch back and provide back the initial target region of interest. So in the beginning, it started around 2 kb, for example, but because it performs short read sequencing, it has to be fragmentized to shorter reads. But at the end of the day, you are getting back the long reads or the reads of interest. This slide is just to show an overview of comparison between Sanger and BTSeq. So Sanger sequencing, as we mentioned just now, is a first generation sequencing where the throughput is considered low because it's focusing on obtaining a single sequence out from a single sample. BTSeq utilizes next generation sequencing systems. So the reads are considered to be high throughput but done in an economical manner, so it can be beneficial to the researcher. In Sanger sequencing, the fragment length is limited to less than 1 kb, while in BTSeq it can go up to 20 kb. In Sanger sequencing, if you want high sequencing accuracy, you need to perform both forward and reverse directional sequencing. But in BTSeq, you just have to perform one sequencing reaction and you're already having high sequencing accuracy. And in Sanger sequencing, you require researchers to provide their own primers for sequencing. But in BTSeq, we do not require the primer information, and if we require, it's just for a validation step. Now, why we do find Salamic's BTSeq technology is a very interesting new technology in the sequencing world, because it comes as a very unique position between the first generation sequencing of Sanger sequencing, while it also in between the conventional NGS method of such long read or short read sequencing platforms, because it does still perform NGS based technology, but it does perform long read sequencing and it does provide high throughput data. But at the end of the day, it comes as a very concise and economical solutions who wants to get the best of both worlds, first and second and third generation sequencing. Now, as I mentioned about the omics world in the previous slides, we also recently started offering metabolomic services, which is one of the upcoming omics field that is getting momentum at the end of as we are progressing in the omics world. So for now, we using our partners from China who has their lab in Singapore, Panomics, we can offer both untargeted metabolomics and targeted metabolomic services. And overall, this is the overview of TREECODE services. And these are the brands we are carrying. Illumina PackBio Salamax is more of the sequencing services that we offer to customers, and the remaining brands are the brands we offer for when it comes to metabolomics and supplying kits, reagents, and lab equipment. So with that, I would like to end my presentation for today, and now we can move on for Q&A sessions. Thank you so much.
Thank you, Dr. Kanagesswari, for your valuable insights. We will now move on to the Q&A session. You are welcome to ask any questions or type them in the chat box, and I will address them one by one. While we proceed, we kindly request all participants to complete the sign-in forms provided in the chat box. There are two forms available, one from IMMB and another from TREECODE. Your cooperation is greatly appreciated. There's one question. Does NGS take time?
Hi. So just to answer about it, okay, if I understand your question correctly, I think you're asking about how long an NGS service takes. So to answer this question, it very much depends on what type of NGS services are you performing at the end of the day. So each NGS services such as whole genome sequencing, transcriptome sequencing, metagenomic sequencing, metatranscriptome sequencing, as well as targeted sequencing, each of them have a different turnaround time, but usually it takes somewhere around 4 to 6 weeks. And it also very much depends whether you want a service provider to provide both the sequencing and the bioinformatics services for you, depending on your project needs. I hope that answers your question.
Any other questions?
So here I'm sharing my personal email as well as our FAS support email, as well as my phone number. Please feel free to contact us if you have any research project that you would like to discuss in further.
We have another question in the chat box. How do you counter misread during sequencing?
How do we counter? So this is actually a very interesting question where I would just like to answer in a more general manner. So each of the sequencing platform has their own platform-based sequencing and nucleotide detection method, right? So we have, as researchers, we do agree that any sequencing platform tend to introduce sometimes sequencing errors. So all of these sequencing errors, depending on how much the sequencing platform is improved to minimize these errors. And also it very much depends on the bioinformatic analysis. So during your bioinformatic analysis steps, you need to take measures to make sure any of the nucleotides incorporated in the reads or your sequencing reads itself of high quality, and you work around filtering out any of these low quality reads. So it actually depends on that. And it's also, once you have the data and you're looking into it, depending on how much publication made available for you and established research made, you can also have a look to understand, is there a place or is there a situation where any of the particular sequence you obtain might not be an accurate sequence that you want. So it's not just at the sequencing level of the sequencing platform. You actually can take a lot of countermeasures during the bioinformatic analysis and during your data interpretation step.
Why does it take 4 to 6 weeks? What is the process that consumes time?
So as I mentioned about the NGS workflow just now, right? Maybe let me just bring up that. So each steps when you're sending to the service lab, they have their own turnaround time and their process to do so. So we cannot exactly say where the steps will take time, but it's just to give an idea, sufficient time to the service lab to perform the service successfully. From where they had obtained the sample, they need to perform some sort of QC to make sure your sample is good enough to perform sequencing. So then they'll perform the library preparation and sequencing and analysis. In my personal opinion, usually the long time or the longer turnaround time is given to focus on the analysis to make sure whatever data you obtain from the sequencing is actually analyzed in the correct or in the accurate manner for us to provide back the results to you. This is just a personal opinion of my personal opinion. So usually it does take quite some time. And okay, again, any service lab that obtain samples from their customers, everything, they have their own standard SOPs of how the service is performed. Of course, anyone who has their own sequencing lab, the turnaround time differ. And like I said, this 4 to 6 is a standard turnaround time I'm giving. Depending on your project needs and what is actually the type of sequencing, it might changes.
If there are no further questions, we will conclude today's session. If you have any additional inquiries, please feel free to reach out to TREECODE. Let's take a group photo. If you have a webcam available, please feel free to turn it on for the picture. Feel free to turn on your webcam for the group photo.
Hello. So I will count like 1, 3, 2, 1. So if you can switch on your webcam, so we can have a group picture. So, all right. So I will just take the pictures. 3, 2. Okay, never mind. Okay, so I will just take the screenshot here. So 3, 2, 1. Okay, smile. 3, 2, 1. 1, 2, 3. 1, 2, 3. Okay, 1, 2, 3. Okay, smile. Okay, thank you. Okay, so yeah, so thank you to Dr. Kanagesswari and also our host here, Miss Aisha, for her assistance to handle this talk. And yeah, so if you have any queries, so you can reach out to us and we will assist you with your your future projects or any questions that you have in your mind. And also just to remind you that maybe you can, for those who are not fill the form yet, so kindly fill in the form. You can get the link in the Zoom, in the chat box. So yeah, so we will end our talk today. And once again, a big thank you to IMMB, especially to Encik Yusri for inviting TREECODE to organize this webinar, and also to our speaker, Dr. Kanagesswari. So we look forward to seeing you in our next session, and hopefully we can work together for your research projects.
Thank you everyone for joining today's session. And I hope you have gained some information from today's session. And once again, thank you to IMBB for allowing us to have the session today. Appreciate it. Thank you. Enjoy your lunch and have a nice day.